Bueno, buenas tardes a todos. Muchas gracias por estar acá. Hoy tenemos la cuarta entrega de Ciencia Humboldt, que es este programa que nosotros tenemos para compartir conocimiento un poco en un tono más científico. Y hoy tengo el gusto de presentar a dos invitadas que conozco desde hace algún tiempo. Alison Schultz, curadora del Museo de Historia Natural del Condado de Los Ángeles, y Rosalind Price, que es estudiante de doctorado en la Universidad de Princeton. Ellas dos son ornitólogas, están con nosotros trabajando aquí con la colección de aves en un proyecto del cual nos van a contar un poco el día de hoy. Entonces, para no extenderme más, los dejo con Alison.
Gracias. Hey. Muchas gracias a todos. Lo siento, pero necesito hablar en inglés. But it's really my great pleasure to be here and tell you a little bit about my research. So my research really stems from understanding the diversity of birds that we can observe just by opening drawers in museum collections. And so there are many ways that we can understand that. In my research, I focus on two main topics. One is plumage color. How and why are birds the colors that they are? It's a really important trait for avian biology. And the other is how are birds evolving due to human-caused changes in the environment? And so today I'm really going to focus on the plumage color side of things. But really the theme of my research is that it uses museum specimens as repositories for many different types of information. So the main group that I focus on for my studies of color are tanagers or Thraupidae, which we can go outside and see if we're lucky and they're in the courtyard at any given time. Colombia has an amazing diversity of tanagers, and what makes them really great for studying many aspects of avian biology is, first of all, they're one of the biggest bird families. Second of all, they're one of the most diverse bird families. They represent every color of the rainbow. It's really fun to make rainbows out of tanager specimens for displays and for exhibits and things like that. They also eat a diversity of food, and they live in pretty much every habitat in Central and South America where they're found. So the first thing that we did to basically study tanagers was during my master's when I was working with Kevin Burns, we made a tanager phylogeny, which enabled us the backbone. We know how things are related to each other, so we can know how they're evolving and map different traits associated with their color. Now, in order to study their color, what we did was we quantified plumage. So basically, we used a device called a reflectance spectrophotometer, which is measuring color from an objective point of view, to quantify color from the avian visual perspective, because birds see differently than we do. For one, birds see into the ultraviolet part of the spectrum, whereas humans, we only see up to 400 nanometers, birds see down to 300 nanometers. And so for each species, what we did, so for example, this is a paradise tanager, beautiful bird. We collected what are called reflectance spectra for each patch. And so you can see for each wavelength, we have how much light is being reflected. Here you can see this is the blue breast, here's the red rump, the green head. And then using a model of the avian visual system, we mapped those in the avian tetrahedral color space. And so each of these represents one of the four bird cones in their eyes. And basically for each spectrum, how stimulated each cone is determining where that point is. But then once these points are mapped in the color space, we can measure things like how much volume are they occupying, how far away are they from each other. And these are measures of basically how colorful is a bird, not just like saying I can look at this bird and be colorful. And just to show you that, for example, paradise tanagers have a very large color volume. They map very, very highly in this measurement of colorfulness that we have. On the other side of things, here's a different tanager, the small ground finch, or Geospiza fuliginosa. You can see that its reflectance spectra are all very flat because it's pretty much a black bird, and all of its points cluster very far together. So this would get a very small score of colorfulness. So it also maps with what we generally think of these birds to be. Now, if we map all of the tanagers in the bird tetrahedral color space, you can see they map virtually virtually the whole thing. They cover quite a lot of overall bird plumage diversity. But one really cool thing is that if we separate these points by females and males, is that females and males virtually cover the whole thing. So we have, very colorful females as well as very colorful males, which isn't true in all birds. And why this is important, because one of the kind of first things that I wanted to study was how is natural and sexual selection shaping bird plumage coloration. So natural selection, say for crypsis, so to blend into the background, versus sexual selection to be more bright, more colorful, so you can attract mates. And how can we study both natural and sexual selection? Well, we make use of the fact that males and females they share the same evolutionary history, the same genetics, basically, but they sometimes have different colors. So for example, in this Rhamphochelys costaricensis, you can see that the male has this very distinctive red and black plumage, where the female has kind of orange and yellow plumage. And so this is, when you think about what this is called sexual dichromatism, where males and females are different colors. Typically, you have a colorful male and more drab or a more cryptic female. However, one nice thing about tanagers is that we have many species where both the males and females are drab, and many species where both the males and females are very colorful as well. And so what we can do is we can study transitions. And really how people used to think of the evolution of sexual dichromatism, we thought that it was males evolving to be more colorful. And so that means that sexual selection is a greater driver. So sexual selection is making males be more colorful. However, an equally possible situation is that the ancestral form has both colorful males and females, and essentially the female is evolving to be more drab or more cryptically colored. And so in this case, you might say that natural selection is the driving force, it's the stronger force. And so because tanagers have this wide variety of species where we have some with, drab, both sexes are drab, both sexes are colorful, and then where the males and females are different colors, we can actually test what's more common. Is it that the male's becoming more colorful or the female becoming less colorful? So let's just do a little cartoon of how we would do this. So here's a little cartoon phylogeny with birds of different colors. One thing we can do is actually to reconstruct what the ancestral colors might have been. And this is using kind of taking into account the avian visual system. And then we can actually measure what's the difference between the ancestral form and the descendant form. And you can see how much evolution would have had to happen to get this descendant form. And we can do that for every branch in the phylogeny to take into account kind of overall phylogenetic relatedness of different species. And do that for both males and females and say, are there more changes happening in males or are there more changes happening in females? Is that distance greater in which sex? And so in tanagers, what we find is that although there are quite a few where both males and females have a lot of change at the same time, the distance is greater for males than for females. So it's actually more likely that males are having more change in their plumage than females are in this group. Now, another thing we might want to do is look at the level of sexual dichromatism. So how much sexual dichromatism high, meaning the differences are very strong between males and females, or low, meaning that males and females are very similar? And is it that colorful males tend to be in cases where the sexual dichromatism is higher? And do we see that same relationship for females? And the answer is that it's really only males where sexual dichromatism is mapped to more colorful plumage. And so what have we found? We found that males have more colorful, they have more change in their plumage. And in species where they're very different, it's that males are more colorful. It's not that females are more drab. And so both of these together suggest that in this group, it's probably, while both sexes are contributing to the evolution of sexual dichromatism, and natural selection and sexual selection are both important, sexual selection is a little bit stronger in driving males to be more colorful than females to be more drab. And now we've been looking at kind of the whole plumage, but what about the different parts of the bird? This is important because different regions of the bird are used more for either signaling to conspecifics, like the crown and the throat and the breast, for example. Birds, when they're communicating, these might be tend to be brighter and more important. Whereas other patches like the back or the primaries or the tail might be more likely to be under natural selection for crypsis, because a predator is probably going to be seeing the back from above, and so they're going to want to blend in. And primaries, those are really important for flight, so birds really can't mess around with their primaries very much. And so what we did was we looked at which sex is driving overall changes in these different regions. We fit a model of either male evolution or female evolution, and then asked, did either males or females fit the model better? And the answer is that for patches that tend to be important in signaling, the male model fit better. So this means that it tends to be, the changes are driven more by males, whereas for most of the other patches that are more important in crypsis or natural selection, both males and females fit equally. Interestingly, for tail, it was a female model. I don't know what that means, but overall, the pattern is that really it seems like it's not just male-driven males driving the evolution of sexual dichromatism, but it's actually in these specific patches that are important for signaling. So it seems like sexual selection is probably driving a lot of the colorful plumage in males that we're seeing in tanagers. And so that's what I've talked to you about now, is how overall color and pattern is evolving. But something I've been thinking about a lot lately is not just, we talk about color as a trait, but color is really made up of a lot of different traits. What do I mean by that? Well, the pattern is made up by the arrangement of the feathers. So, where are the feathers on the bird? In the last slide, we saw that actually where on the bird we're talking about matters. But then each feather is made up by macrostructures, so kind of the overall shape and dimensions of the feather. And then we can zoom in and look at the microstructure of the feather, which is made up by barbs and barbules. We can zoom in even more and look at the nanostructure, how the molecules are arranged, and then how the different molecular composition. And so when we're talking about color, we're really talking about all of these different things that can be varying on birds added up to produce one phenotype. And so one thing I'm wondering is, how does the variation in these different levels restrict the possibilities for birds to be certain colors or certain patterns? And so that's what I've been thinking a lot about in my research lately. And so just for those of you who don't think about bird colors all the time, which is perhaps some of you, we typically in the field think of birds as being colored by either pigments. Pigments absorb certain types of light. The most common ones are melanins, which produce blacks, browns, and kind of what we call rufous colors, this kind of orangey color. Or carotenoids, which produces most of the reds, oranges, and yellows in birds. Those are very common pigments. Carotenoids are thought to be really important for sexual selection as indicators of a bird's quality, because birds have to eat carotenoids in order to put them in their feathers, and they also have to metabolize them sometimes. But then also we think about colors, all the blues, the greens, UV colors, iridescent colors where changes by angle are produced by the nanostructure. So how the molecules are arranged and how they're refracting light. But one thing that we've been thinking about lately is how microstructure. And so what really got me thinking about this in my own research is this bird, Ramphichelys carbo. And when I had been measuring this for my tanagers, this bird almost always measured as black instead of measuring as red, as a deep maroon color, which I could clearly see with my eyes, but its reflectance was very low. That had always puzzled me until another grad student, I was a grad student at the time, published this paper on what's called super black plumage. And what this means is that the arrangement of the barbs and the barbules are actually absorbing more light than the pigments alone. And so in this case, this is kind of a normal black feather. This is the barb, these are the barbules. And when you're getting it ready for scanning electron microscopy, you do what's called sputter coating. You cover it in gold. So you can see this black feather turns gold, But in this super black feather, you can see that the barbules are very different shapes. Even after you cover it in gold, it still looks black. So this feather is just absorbing more light. And so that kind of got me thinking, does Ramphichelys maybe have this different microstructure that's absorbing more light and giving this kind of maroon appearance? So I asked Cody this question, and Cody thought it was also interesting. Before I talk about Ramphichelys more, I just want to, for those of you who don't think about feathers all the time, some of the terminology I'm going to use. So here's your typical contour feather or body feather. We have the pennaceous part, which typically has the colorful part of the feather, the plumaceous part, which is the downy part. Off of the central rachis, we have what are called barbs. If we zoom in on those, each barb has a central shaft, which is called the ramus. And then off of that are these little barbules. And so mostly what I'm going to be talking about is the morphology of the barbs and the barbules. That's going to be varying. So we decided to look at both the carotenoid pigments, because again, carotenoids are thought to be important in sexual selection, and the microstructure across Ramphichelys tanagers, which are 10 species. We used LCMS to look at the chemical composition, and then scanning electron microscopy to look at the structure of the feather. And all of this research was possible because these specimens were available at the Museum of Comparative Zoology at Harvard. And so the first thing that we found is that the carotenoids are the same between males and females. So even though they look different, the types of carotenoids that are there, and even the amounts of carotenoids, are significantly correlated with each other. This was very surprising, because if carotenoids are supposed to be importance of sexual selection, and we have these really bright, colorful male tanagers, we expected that carotenoids would be different. So carotenoids are the same. What about the microstructure? Well, the next thing that we found was that there were several different types of microstructure. So this is kind of your typical female feather. This is a cross section. Here's the barb, here's the barbule. You see the barbules are flat. But we found males actually had many different types of color. So in bright red feathers, they might have an enlarged barb with kind of angled barbules that are small. In Ramphichelys carbo had what we thought, which was angled barbules, which are giving it that kind of super black structure, but with carotenoids inside. And then we also found some red feathers that have these really oblong barbs and angled barbules. And then if we plot males versus females, you can see males occupy a much greater variation in these shapes of the barbs and barbules than females, which all kind of cluster together here. We also did some simulations just to ask, how does varying this morphology actually change what light is coming off of the feather? This is just changing the angle of the barbules. So again, here's the barb and here's the barbules. If we change the angle up, starting at about 40 degrees, which is actually where the barbules are on the bird, reflectance really drops down. So the angle of the barbules alone does change how much light is coming off of them. And then if we kind of make the barb itself kind of more fatter and more oblong, we see how much light is coming off of it. More light is coming off of it when it's kind of bigger and fatter, just like those red barbs that we see in Ramphichelys tanagers. So what did we find? This is all very, very exciting. We found that actually microstructure had a greater impact on the optical properties of the feather than the carotenoids did alone. And they're really different between males and females. And again, surprisingly, the carotenoids didn't vary between the sexes. And so, what this has really made me think about is the fact that we've been focusing a lot in our research on the pattern. We've focused a lot on the nanostructures and the molecular composition, but we really need to be thinking about these other axes of diversity in feathers, the macrostructure and the microstructure, when we're thinking about our research and trying to understand how are colors evolving in birds. Another thing that I've been thinking a lot about lately is feather functions. So, I've talked a lot about color as basically a communication means, but feathers and colors have many different functions. We recently had a review with Ryan Terrell. This was actually just accepted. We found 26 different things that feathers do. And so when we're thinking about how is color evolving, we really need to be thinking about not just color as a signal, but color for thermoregulation, for protection from solar radiation, for protection from parasites. all of these different things that might also be driving the evolution beyond just being cryptic or being, more brighter to attract conspecifics. Just for fun, what we did was we actually mapped these different functions on either what type of the bird have they been thought to be from, and what we're calling hierarchical level. So what aspect of the feather have they been tied to? Just so, for example, so color and pattern, you can see these red are different types of communication functions that have really been associated with the head, with the ventral side, with the dorsal side, whereas flight and locomotion has been associated with the wing feathers. And we did this for all of the different hierarchies, and you can see there's a lot of blank spaces. And so really, as researchers, we need to understand which parts of the bird, which parts of the feather, which parts of the bird are associated with these different functions. We can understand how is selection able to kind of balance multifunctionality of feathers. And so I've told you a little bit about my research. Rosalind's going to take over from here, but one thing I just wanted to emphasize, you are all collections people, so you're aware of the value of collections, but none of the research that I presented would be able to be done without museum collections, because, they're really a library for having, not just the colors, but also having the feathers available. And we never know what types of research we might want to do with museum specimens in the future. So we have to keep collecting them, and we have to keep preserving them, and every part is valuable. So with that, I'm going to turn it over to Rosalind, who's going to talk about her research and some of our joint research.
All right, well, thank you all so much for being here, and it's really nice to meet you. I'm Rosalind. So like Allison, I'm very interested in color evolution in birds and color perception in birds, and particularly in the genetic and physical origins of variation in color. So I started by exploring the genetics of color through this review paper that I wrote with my advisor, Cassie Stoddard. And this review was really organized around recent advances and emerging questions in avian color genetics, including the genetics of colors in feathers, in bear parts, and in eggs. And I'm going to tell you about some of my favorite sort of takeaways from the review, but I just want to highlight that when we published the review, the cover image for the Journal of Heredity was this really, really beautiful photo of eggs taken by David here in the collections at Humboldt. So yeah, we were really excited about that. So like Allison mentioned earlier, birds have a lot of color diversity that's produced using various pigments and structural colors. But in spite of all of this color diversity, until recently, only the genetics of melanin pigments were really well known. So melanins, like Allison mentioned, produced browns and blacks and some orange colors. So this really barely scratches the surface of avian color diversity. But there's been a lot of recent progress in color genetics, particularly in the genetics of carotenoid pigments, which I'm going to tell you just a little about today. So carotenoids are these pigments that produce most of the yellows and oranges and reds in birds, and they're the second most common pigment after melanins. But birds can't actually synthesize carotenoids, so they have to get them from dietary sources. All dietary carotenoids in birds are yellow, and they have to be metabolically converted into red. So one open question for a long time has been the genetic and molecular basis of this conversion. And recent work in zebra finches and canaries mapped this yellow to red conversion to a particular enzyme called CYP2J19. And since then, there have been a lot of studies in all kinds of different birds that have sort of pointed to the same enzyme as being really important. And actually, since we published this review like 4 days ago or something, there was this other really amazing paper that was published that shows that this other enzyme, DDH1L, is also probably super important for this conversion from yellow to red. So altogether, this is just like a pretty exciting step forward in the genetics of coloration in birds. But one thing I think is really cool about this genetic story is that there are some pretty clear links to the visual system. So color vision in birds is mediated by cone photoreceptors in the retina, and birds are tetrachromatic, which means that they have 4 kinds of cone photoreceptors, which I've drawn here. So each of these cones is paired with an oil droplet, and 3 of the 4 oil droplets also have carotenoid pigments in them. So the function of these pigments is that they filter incoming light, which narrows the spectral sensitivity curves of the different photoreceptors and means that color discrimination is a little bit higher, although that's at the expense of absolute sensitivity to light. So if we zoom in on the medium and long wavelength sensitive cones, you can see that one has an oil droplet pigmented with yellow carotenoids and the other has red carotenoids. So just like in feathers, the yellow pigment is converted to red with this enzyme, CYP2J19. And after this enzyme was first identified in feathers, there was a few comparative studies that looked at this enzyme more broadly. And they found that this enzyme is actually present in both birds and turtles, where it plays a role in pigmenting those red oil droplets. So these droplets are nearly ubiquitous across birds, so almost all birds have them, but only a few birds have red feathers. So that really suggests that the production of carotenoids evolved in the context of the visual system first, before being co-opted for color in skin and in feathers. So in other words, birds could actually see red colors before they were able to produce them in their feathers, which is pretty cool. Okay, so there are all these sort of recent exciting advances in color genetics. And I just talked about one of my favorite ones here. But overall, I do want to highlight that there's a lot of avian color diversity about which we know almost nothing about the genetics. So for example, in this first row up here, we have a lot of pigments that are sort of unusual or taxonomically restricted, and we don't know a lot about those. Bare parts, as opposed to feathers, are really important sources of colors in birds, but we generally know more about the genetics of feathers than about the genetics of bare parts like eyes and bills and skin. And then finally, structural colors, which are a huge part of color diversity, and I think are really beautiful. We just don't know as much about the genetics of those colors, although there's a lot of active research in that. All right, so in this next part of the talk, I want to turn to the project that actually brought us here to Colombia to collaborate with Gustavo and the ornithology team. So this project focuses on physical mechanisms of color production, and specifically the role of microstructural modifications in structurally colored and pigmented feathers in Tangara tanagers. So earlier, Allison introduced you to the idea that changes in microstructures of feathers can change the appearance of how these feathers actually look, as in the case of those superblock feathers or in these red feathers in Ramphichelys tanagers. So these examples are really exciting and fascinating. But one big gap is that we don't really know how important these microstructural modifications are in color space in general, particularly in structural colors. So in this study, we really wanted to understand how widespread these modifications might be and how important they are for color production across color space, including in pigmented feathers and in structurally colored feathers. And we're also really interested in how microstructural modifications might be different in different parts of the body. And that's because microstructures are also important for non-signaling functions like water repellency or thermoregulation or flight. So we expect that there might be some sorts of trade-offs between color production and these other functions that might constrain microstructure evolution. So we chose to study these questions in the tanager genus Tangara. And as you all know very well, these tanagers have really spectacular color diversity, which includes colors produced by structural colors, by pigments, and by interactions between pigments and structural colors. So for this project, we sampled feathers from 26 out of 27 Tangara species from museum collections. And we took feathers from 7 standard regions, including some regions that we think are more important for signaling and some regions that we think might be less important for signaling. And in addition to tanagers, we've also sampled other lineages in the bird phylogeny that represent independent origins of carotenoid pigmentation and structural colors, which are shown by the red and blue squares in this phylogeny on the right. So for each of these specimens, we collected a range of data to characterize the feather color and relate it to microstructural characteristics of the feather. So we first measured color from specimens using UV-Vis reflectance spectrophotometry, like Allison talked about before. Then we took a single feather from each patch of interest. And we imaged the dorsal and ventral surfaces of the feathers using scanning electron microscopy. So here's a SEM image of the crown of this Tangara chilensis. We also imaged the feathers using confocal microscopy. So this was a way of getting really high resolution images of the 2D features of the feather in color. And then one other really important piece was actually understanding the shape of the barb and how pigments and nanostructures were arranged within the barb. So we cross-sectioned these feathers and then we imaged them again in this confocal microscope. So basically here you can see this blue area on the bottom, that's structural color. And on top of it is carotenoid pigment, that's yellow. So these together produce this green color. And then finally, we took these cross sections and we imaged them in the SEM to really look at the nanostructures and make sure that we were sort of understanding what was happening in these feathers pretty well. So once we had all of these images, we measured characteristics of the barbs and barbules using the program ImageJ. We sort of separated our feathers by their underlying color production mechanisms. And then we extracted measures of brightness and saturation from our spectral data. So altogether, we had like a lot of different measurements describing the 2 and 3D shapes of the feathers, the relationships to pigments and nanostructures within the barbs, and then the color of the patch. And we analyzed these data using phylogenetic comparative methods on a couple of different phylogenies. So in this first part, I'm going to tell you about how microstructural modifications in these Tangara feathers are really widespread and associated with novel optical effects. So the first big result is that there's a ton of variation in barb width based on the underlying color production mechanism. So on the right, there are a couple images of feathers with the arrows pointing to the barbs. And then on the other side, I've plotted the width of the barb at 3 different points. So sort of at the base, midway, and at the tip. And you can see that the red, blue, and green feathers have really wide barbs that become up to 3 times as wide in the colorful part of the feather, while these black melanin-containing feathers have narrow barbs that don't really change in diameter at all. And along with these changes in barb width, there are also some big changes in barbule density, including a lot of feathers that actually appear to lack barbules completely. So in general, red, green, and blue feathers tend to have reduced barbule density, while black feathers tend to have a much higher barbule density that doesn't really change a lot along the barb. So overall, what we end up with is this really pronounced and striking axis of morphological variation, where on one end we have feathers that have these small barbs and long and dense and thick barbules. And on the other end, we have feathers with thick barbs and short, thin, sparse, or even no barbules. And these barbs and barbules kind of vary inversely, so that wider barbs have fewer barbules and vice versa. And I just plotted this qualitatively here because I want you to actually like be able to look at the feather, but this also falls out pretty nicely on a PCA. So given all of this sort of variation in these feathers, the next big question is, how does this variation correlate with different optical parameters, especially the brightness of the feather? So as you might expect, there's a relationship with microstructures and brightness. So I plotted brightness against the microstructure principal component 1 here, and you can see that the wider barbs with smaller barbules are much brighter than the other end. So this really indicates that feather brightness is really changing as a result of microstructural modifications and not just changes in the underlying pigments or nanostructures. There's also a lot of variation in microstructure across the body, which I've plotted here for structurally colored and structure pigment feathers. So for example, you can see that structurally colored breast and epaulet feathers tend to have wider barbs and fewer barbules in the breast and epaulet than in the belly. And these patterns don't follow what we would expect if these microstructures were just scaling with feather size, for example. And then on this other side is sort of an individual example with a familiar bird. Here's an emerald tanager, which has feathers that are, or that include both pigments and structural colors. So this bird is like green all over, but you look at the belly and you can see it has these pretty thin barbs with lots of barbules, whereas the crown has these really thick barbs that lack barbules completely. So overall, this suggests that microstructural modifications are being shaped by competing demands for color production and non-signaling functions. But there's also a lot of spread in these data that I think we're excited to sort of dig into in future projects. So up until now, I've just been talking about Tangara because they're the focus of this work. But we've also looked pretty broadly across birds, and we're seeing very convergent effects in all of these different lineages where there are modifications such as reduced barbules and wider barbs in really brightly colored feathers. So this leads to some pretty interesting questions, not only about signal evolution, but about the developmental and genetic mechanisms that produce these modifications in distantly related birds. All right, so I've just spent a while telling you about how barbules can be reduced and how that affects feather brightness. So in this next part of the talk, I want to tell you about one really sort of fascinating twist to this, which is that barbule loss appears to happen through different developmental mechanisms in these 2 types of feathers. So we know that barbs that lack barbules have brighter or are associated with brighter colors. But how does this actually happen developmentally? How do these feathers get rid of barbules? There are maybe a couple of ways this could happen. So for example, you could just not develop barbules at all. Another strategy might be hiding the barbules. And it turns out that carotenoid feathers and structurally colored feathers are actually using both of these strategies. So if you look at the carotenoid feathers, you can look at the dorsal or the ventral sides, and you don't see any barbules. So what happens here is that the barbules are just not branching off from the barbs during feather development at all. But if you look at the structurally colored feathers, it turns out that they do still have barbules, but they're hidden on the ventral side of the feather. So over here is, on the right is a colorful feather, the dorsal side. You don't see barbules. But if you flip it over and look at it in the SEM or another microscope, you can see all these tiny little barbules hidden underneath. And we've seen this pattern sort of overwhelmingly in our data. So that really suggests that structurally colored feathers are doing something totally different than carotenoids to hide their barbules, and that these two types of feathers have independently evolved this mechanism to become brighter through different developmental mechanisms. So you might be wondering, what about feathers that have both structural colors and pigments, like many green feathers? So when we look at these under the microscope, they appear to be sort of intermediate. So they do have ventral barbules, but not as many, and the barbules are a little smaller. So that suggests to us that there's a relationship between the amount of carotenoid pigment in the feather and the degree of ventral barbule differentiation. So to further explore this hypothesis, we turn to these cross sections of the barbs. And to orient you here, the glowing blue parts are all structural colors, and then you can see red or yellow pigment on top of that. And one thing that really jumps out about these cross sections is that there doesn't appear to be a lot of internal differentiation in the carotenoid feathers. So for example, many feathers often have like an internal vacuole or something, and these feathers don't have that. So that really suggests that feathers with more pigment in them have a lack of both external and internal differentiation, which again suggests that there's a relationship between carotenoid content and feather development. And this relationship holds up when we test it statistically. And there have been a few studies that have suggested that carotenoid deposition into developing feathers does impact the structure, the branching structure. So we were really interested to see that in our data and really excited to see how this interacts with structural colors in feathers. All right, so overall, microstructural modifications are a pretty important part of color production in both structurally colored and pigmented feathers and associated with changes in brightness. These effects are taxonomically widespread in tanagers and in other birds, more extreme signaling patches, and likely the result of alternate developmental mechanisms in structurally colored and pigmented feathers. So this is exciting, but I do want to highlight that we really don't know a lot mechanistically about how this happens developmentally or genetically. And so that is really what has brought us here today. So for the next couple of weeks, we're going to be collaborating with Gustavo and a lot of you all. And the plan is to go into the field for a couple of weeks. And what we're hoping to do is sample pin feathers from molting tanagers. Probably not Atenga argilensis, but other ones. And from these pin feathers, we can actually build developmental time series. And that's because feathers grow from the base. So the tip of the feather actually ends up being older than the base of the feather. So if you take one of these pin feathers and you cut it at different sections, you can actually have sort of a time series that tells you about the different developmental events that are leading to these different kinds of feathers that we're seeing. And we're also collaborating for this part with Choming Chong and Ping Wu, who are at the University of Southern California and are at the Lab of Tissue Development and Regeneration. Okay, so with that, thank you all so much. We're really, really excited to be here, and we're excited for the chance to work with many of you in the field for the next couple of weeks. And with that, yeah, we'll thank all our funding sources and collaborators, and I think we'll take any questions.
Si alguien tiene preguntas. ¿Cómo?
¿Español o inglés?
Como quieras.
¿En español o en inglés? ¿Español? Despacio, listo. Súper interesante porque casi siempre cuando, yo no estudio aves, pero casi siempre ve uno estudios asociados a colibrís en toda la parte estructural del plumaje. Entonces, claro, cuando piensas en tángaras son súper coloridas y no había visto estudios en ese grupo. Esto de las microestructuras también ha sido estudiado en colibrís, en hummingbirds, y se ha visto algo similar a lo que ustedes están encontrando, o no se sabe mucho de las microestructuras cuando hablamos de qué color estamos viendo nosotros y qué color es reflejado en ese grupo.
Is it okay if I answer in English? Okay, thank you. That's a great question. All right, so hummingbirds are interesting and very beautiful, but they are a little bit different because they make their structural colors in the barbules. So iridescent structural colors, we didn't mention at all because they're really complicated. They're produced by these nanostructures of these organelles called melanosomes that are in layers within the barbules. And so the structural colors we study, these are all in the feather barbs. And so the changes that we see are pretty, sort of simple. It's like the barb gets bigger or smaller. But as far as the microstructures of the barbules and iridescence, that's a pretty open area of research that a few other people are studying. But some examples that are out there, for example, are like hummingbirds have these barbules that are shaped sort of like V's, if you look at them, or there are really interesting examples from birds of paradise where their iridescent barbules also undergo microstructural modifications. So the answer to your question really is that I think there are likely really, really interesting things that are happening with barbules in iridescent birds. But because the underlying structural color develops so differently than tanagers, it's really like a whole different question. And the physics are very complicated as well. Anything to add to that, Ellison?
I just say, microstructures have been largely ignored in most birds for a really long time. So we don't know much about it in anything, which is exciting. It means there's lots to do.
Yeah, but hummingbirds, I think, are an amazing system, and I hope that people are studying it.
Yeah.
Muchas gracias por la presentación, fue muy interesante. Cuando ustedes hablan de que el alimento puede estar alterando el color en las aves, ¿se refieren a qué? ¿A plantas, a insectos? Y si es a plantas, ¿qué tipo de plantas generan esa variación del rojo o del amarillo al rojo, como creo que lo hablaron ahorita?
Did you get it? What kind of plants cause that variation?
What kinds of plants?
Yeah, in diet base change.
Okay, yeah.
What kind of plants?
Okay. Well, I'll start with this and you can finish it. So the key for, especially carotenoids is that they have to be gotten from the diet. But there are many, fruits, of course, have a lot of carotenoids in them, but also insects have a lot of carotenoids in them as well. You think of actually the classic example of carotenoids are flamingos, which get them from shrimps. So birds can get their carotenoids from many different parts of their diet. It's actually the importance of carotenoids as being an honest indicator. It used to be that people thought a bird was a better forager when they had brighter carotenoids because they could eat more. And now it's thought to be much more complicated. And I actually think that there's a huge bias in a lot of people have studied the importance of carotenoids from the temperate zone, temperate birds perspective. But, if we look in the tropics, actually many things have carotenoids. And a lot of times, for example, we'll have blue males and green females. So it's the females are adding carotenoids to make themselves more cryptic, which suggests that carotenoids are not a limiting factor in the diet, actually. So, which plants they are really will depend on which species we're talking about. But I think carotenoids are much more widely available in the diet than kind of has traditionally been thought.
Yeah, the only thing I'll add to that is that even once carotenoids are ingested by the birds, there are still lots of different transformations that happen. to make this color diversity. So, I talked about this yellow to red transformation in the carotenoids, but also birds can deposit different kinds of carotenoids in the same feather. Oftentimes the carotenoid molecules undergo sort of conformational changes by binding with the feather keratin that can also change the colors. So it's like really this whole palette of these carotenoids that come from plants that can then be changed even more.
Muchas gracias por la presentación, de verdad, estuvo muy interesante. Cuando veía los materiales, yo me preguntaba como cuál es la implicación en el calor también, ¿cierto? Y si de pronto lo han pensado en algún momento, porque pues uno esperaría que un material súper negro como estos colores absorbiera más calor. ¿Han evaluado algo al respecto?
Did you get that? Implications of super black in thermoregulation.
I think that's a really great question, and I think we don't know the answer, but it's a question that I am very interested in asking. I'm actually hoping to get a spec that can take infrared measurements as well as color measurements and want to look at super black feathers. One thing that they have looked at are melanin pigments, carotenoid pigments, or iridescence, and how that impacts potential for thermoregulation. And they found that actually iridescent feathers absorb more infrared radiation, so more heat than other types of feathers. That's thought to be due to all the melanin that's in the iridescent, so it's a little bit complicated. But I think the question that you're asking is like, what are the other functional implications for these microstructural modifications is really, really important, and something that we're hoping to think about in the future, and something that I think you all are, being positioned where these birds are actually living are well posed to think about and answer as well with fieldwork. You want to add anything?
¿Alguien más? Dani.
No, yo les quería preguntar si digamos que la preparación de las aves, ¿cierto? O sea, la forma en la que yo preparo aves de alguna forma puede afectar esas microestructuras que ustedes están viendo, o es algo que, o sea, que no depende mucho de la preparación de las plumas y esas cosas.
Okay. You start and I'll jump in.
All right. Well, Allison, as curator, is probably better positioned to answer that one. But I think if you're asking about how the preparation of the specimen really affects the color, we don't think that color degrades a lot in specimens, or that the microstructures really change over time. And so with this project, we were careful to get specimens that were, sort of as recent as we could. And we tried to get them all from the breeding season and so on. But in other studies, like the one that Allison talked about with Ramphichelys, there don't appear to be any effects on specimen age and color characteristics. And we're really excited, to be accessing really fresh feathers on this trip. So just to make ourselves feel better about this question, we're planning to look at some of those feathers and make sure that we're not seeing big effects of age of the specimen on these microstructures. But, it's like they're dead keratin. If the specimens are sort of carefully preserved, then it shouldn't really change the microstructure a lot.
Yeah, I'll just add, there have been studies that have shown that melanin can fade over time, as well as carotenoids. So pigments can potentially fade. Partially depends on how well the specimen is stored. So it's stored in good cabinets, protected from light. Probably it's not going to change a lot. If you're looking across species, the differences are probably minimal. But if you're looking at like how a specimen is changing with, small geographic distances or through time, that's when you might be a little bit more worried about, some of the effects of specimen preparation. One thing I'm actually very curious about, which I don't think anyone's looked at, is how preparing fluid specimens changes feathers. So if you stick something in ethanol, does that change the conformation of the feathers as it's becoming more common? So I think that's a fruitful thing to look at, would be to compare feather structure in fluid preserved specimens.
Bueno, muchas gracias, estuvo muy chévere. Quería saber si ustedes han pensado cómo qué trade-offs puede tener para la pluma y para el ave tener menos bárbulas y barbas más gruesas.
Go ahead.
That's a great question. We definitely have thought about it a lot without coming to a clear answer. But basically, you know, barbules are very important for a lot of these functions. Like water repellency is a really big thing for barbules, and thermoregulation as well, and of course flight. And so, you know, Allison mentioned earlier about flight feathers, how they generally are not that colorful because there are these constraints on them. And I didn't present this in this part of the talk, but you know, we also looked at primary feathers, and they always have barbules, always. So that, you know, is a pretty clear example of how you cannot make these wing feathers so colorful, because you need those barbules to be hooking together for flight. And, you know, that emerald tanager example that I showed as well, it's like, all right, so maybe the crown has no barbules, but that's much harder for like a belly feather because there are all these pressures for thermoregulation. So, you know, the sort of vague answer is that these trade-offs definitely exist, and we're thinking about them a lot, and we'd love to keep talking about them. But as far as being able to say, like, this is exactly the story that's happening, that's a little harder at this point, particularly without doing clear experiments on like, how does getting rid of barbules make the bird, you know, more susceptible to water or something like that? Anything else?
Yeah, I'll just, you know, that if you remember my giant matrix of functions by different parts of the feather and different parts of the body, I think really what we need to do is make that matrix for different species, and then you can correlate different life histories. Like, okay, if a bird lives in an open habitat, it can have variation in, you know, the ventral side, but not the dorsal side in these parts of the feathers. And also why the developmental aspect is important, because let's say the macrostructure, the proportion of the downy feather versus the colorful feather might be important for thermoregulation. But let's say the, you know, water repellency is driven by the microstructure. You can start to understand how, because these different parts of the feather and different parts of the body can be under different selective pressures, that can help these feathers to be under different types of selective pressure. But if they're developmentally linked, like having pigments versus the microstructure, then birds are constrained to have, you know, then they're not independently, they can't be independently driven by different types of selective pressures. If that makes sense. They're constrained in that way.
Bueno, tengo entendido que la pluma es una estructura muy antigua. Es decir, se habla como de fósiles, se han encontrado como estos dinosaurios con plumas y todo esto. Entonces la pregunta es más como hacia, teniendo en cuenta la evolución de las plumas como estructura y la evolución de los pigmentos, si se sabe cuál de estas dos, si la microestructura o el pigmento, cuál empezó a tener primero influencia en la coloración.
Sure. Okay. 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 So just to make sure I've got it right, you're asking which came first, the microstructural changes or the, I would say, well, okay, it's probably complicated, but I would say in general, probably the underlying pigment evolution came first. And that, you know, is something we can sort of see if we look, you know, in particular systems, it's like, you can probably trace the evolution of the pigments coming and then sort of inducing these developmental changes. But it's also complicated in that I think also likely, how do I say this? I can't even say this in English, much less in Spanish, but Yeah, I would say if we look sort of broadly, it's likely that the underlying pigments like melanin or carotenoids evolved first, and then these more extreme microstructural morphologies like super black or something, those came later to enhance the underlying effect. But I do think there are pretty deep developmental links between the carotenoid deposition and the microstructural changes. Or with the structurally colored feathers, I think those things probably happen pretty quickly together.
Yeah, I agree with that. And then I just, one thing that we're starting to do is look at we've been focusing on very colorful birds for all of ours. Ramphichelys and Tangara are both very colorful clades. So branching, kind of extending out and looking at less colorful birds and seeing what types of microstructures they have is kind of the next step to start to trace these kind of evolutionary histories. So how much carotenoid do you need in order to start to induce these microstructural changes is, what appears first on the tree. Hopefully we'll be able to answer that sometime soon.
¿Alguien más? Bueno, entonces muchas gracias, Alison, Rosalind. Fascinante. Listo, Diego, sigo por ahí.